My role here is a very brief one to introduce the three speakers today. These guys are all very talented and hard-working people who are going to present to you on three quite different processes and projects to show some of the journey to create a cell therapy manufacturing system. So I guess to set the scene for that sort of work, when we look at a process and a manufacturing system and think about what we might do in partnership with our clients, we go into the company and we ask a lot of questions about the processes that they have at the moment. So we start off saying, well, what goals do you want from a manufacturing system? It needs to be affordable, needs to have a robust, consistent, repeatable process. Do we want it to be functionally closed for process integrity? Where's our process now? It's likely to be a manual process done by highly skilled operators in an open form, in a biosafety cabinet in a clean room. Where do we need to be? What sort of process development has to happen to take that process to something that is affordable and consistent and repeatable? What are the nice to haves? What are the must haves? The outputs of such work might lead you to a phased development plan where your process development people need to make some modifications to the process to fit with the automation and the disposables for functionally closing the process. And but you may decide not to do all of those activities in the first or in one big project. You might spend the capital, do the development to get it to a stage where it's suitable for commercial or for phase 3 or early commercial production before investing in the dollars to take it the whole way. So at the end of this initial engagement, you end up with a capital plan, a time development plan, and a process development plan that will take you on that journey. So the steps are review the process, decide the path, test alternatives, develop solutions, and then fold those into a production environment. So initially I'm going to introduce Dr. Ian Harris, who's the Senior Director of R&D at Janssen.
So to give you a general overview of the product we're developing, it's called Center 2476. It's an allogeneic somatic cell therapy for the treatment of retinal degeneration. We're currently in clinical trials for geographic atrophy, which is the severe form of dry AMD. In terms of the high-level CMC goals, we are manufacturing 4 different doses with a 10-fold range, and all those doses are to be cryopreserved and meet stability for the entire time of the clinical trials. The product is ready to inject, meaning that after it's been shipped to the clinical site, it's thawed and aspirated and delivered through a microcatheter. So that's the only handling steps. There's no processing in terms of reformulating or diluting. The important criteria for us is delivering quality product to patients through the clinical development and commercialization of the product. In terms of our strategy, we really do need to balance the technical risks, comparability, and also our expenditure into the process. So we want to make timed investments so that most of the investment in our CMC equipment and facilities is occurring after the clinical proof of concept. So what's shown along the bottom here is our equipment trained for phase 2B. Hopefully next year I'll be able to show you the equipment for phase 3 in commercial and show you how for each unit operation that's evolved. One of the key things that we've looked at is the manufacturability, and having the input of experienced GMP operations in the design of the equipment and the processes is very important. So in terms of manufacturability, one of the key things is that can you validate the process? So for example, the manipulation of cell cubes, and the steps in formulation we thought were key to our program, and being able to validate that was essential. Also minimizing failures and deviations in our process, and we see that automation can really address those needs. What's often not talked about is the training and qualification of operators, and we really want to minimize that expense.
Argos is a fully autologous cell therapy company. Some of the key attributes of our technology, and I'll talk about it in more detail in the next few slides, is that it targets the mutated antigens in the patient's tumor. It ignores the non-mutated antigens. The technology overcomes immune suppression that exists in cancer, and we also do infectious diseases. And I'll talk about the mechanism, or at least our proposed mechanism of action, but it induces CD8, CD28 memory T cells, and there's no PD-1 checkpoint inhibition. We have a cost-effective, scalable, automated, and centralized manufacturing process that we've collaborated with Invetech on, I guess, for a decade. Although we had a break in the middle, it wasn't a full decade. And it's a platform technology, so it's applicable to really all cancers where you can isolate tumor, and also with infectious diseases. Now I'm going to go really into the talk Richard told me to give and talk about the commercialization challenges. And we've talked a little bit about logistics, both for the patient samples and the final sub-product. I've already discussed that. Throughput, you've heard this probably several times yesterday. It's really scale out. They're individual personal products. So it's not a scale up, it's not a reactor. You need to go, for instance, on kidney cancer, our projected peak volume is going to be about 10,000 patients per year. So you have to figure out a way to scale this process out. You need a good cost of goods. I mean, if you do this manually, the labor involved is the bulk of it. The cost of goods and the pricing go together. It depends on the clinical efficacy, how you price it, but you'd like pricing flexibility, and I believe automation gives us that pricing flexibility. What I really want to point out, a dramatic reduction from the manual to the automated process and labor. We don't show the facility there. These other costs, reagents and disposables. Disposables are relatively low on the manual end. They're going to be a larger cost, but the areas, the point of these two pie charts is the area of these circles and pie charts are proportional to the cost. So think of it, you're roughly 5 to 6 times lower in cost when you move from a manual process to an automated process. We started work with Invetech, and we had what we call alpha prototypes for both the RNA and the cellular delivered really within 2 years from the project startup.
So what's immediately clear from the picture here is that our product is a little bit different from the last two, from what Ian and Fred had discussed. Firsthand you can see it without a microscope, and actually there's no cells in this final product. It takes cells to get there, but at the end of the day there are no cells. So this is a picture of the investigational bioengineered vessel. We've got 2 different indications that we are currently in human trials for. One is for peripheral artery disease, and the other is for end-stage renal disease. One of the keys that we're kind of living by is you want to translate the process from the laboratory to be a robust manufacturing system with your eyes on commercial scale production and making high quality product and giving yourself the best path and the best your best chance of regulatory approval as well. We started with our design. Design is based on commercial scale production and in making sure we embed quality into the process. But from that design, we do some basic testing, as inexpensive as we can make it, sort of a unit ops test just to show feasibility of the actual design. And then moving from there to a pilot scale, from the pilot scale onto a full-scale R&D process run. So I think what's been key for us is really prioritization of this list of technical hurdles that we have in front of us based on risk, based on cost, resource availability, and you know, the strategy of moving from design to a unit ops test to pilot scale and so on really helps address some of the cost issues. You can vet out your design quite a bit before you move forward with it.
Can you guys talk a little bit about the software elements of the systems that you've developed?
The systems that we design are actually pieces of industrial equipment, so they're using PLC software. So we're designing the software to run the process. So we go in there and we draw out in detail the process steps, the times, the temperatures, and we build that into the PLC code that runs the equipment. And as I say to Lee Williams at Argos, who owns that R&D device, you know, the robot makes the same mistake every time. And once you eliminate the mistakes from its repertoire, it does the same process. So the consistency of output is amazingly solid.
How do we feel this sort of, or fill that valley of death between clinical data, automation, and BLA?
The first thing that we do, as I said in my introduction, is come in, do a deep dive on the process, try and understand the process and pick the most appropriate technologies for taking that forward. That can develop a staged capital plan that matches your process development plan. Now, but it costs, like I said, about $150,000 to do that first study, but it gives you a plan that you can then use with your investors as a path forward. They know how long it's going to take to get into production, what steps have to go through, and then you can make a financial decision on that. We find, and I'm sure that Fred can talk to that some of the material that we produce in the work that we do is very persuasive for the investors in encouraging them that there is a path forward.
It has to be done early. The stories I hear about, Dendreon is if you wait too long, you kind of get fixed. You get stuck in the mud on yourself too. You run out of time, you get stuck in the mud, and God bless them for getting approved, but now they got this other problem that's really hard to solve when you're commercial. So Dr. Healy, this is the conundrum. I don't really have a good answer. You got to do it early enough. You wait. We're actually crunched now because we put automation on the back burner for a while after this Kieran relationship broke up. So now we're trying to cram. I don't want to say cram, but it feels like that, right? We're trying to finish this project off at the time we're going to get the data for the phase 3. And God help us if we don't, because the worst thing is you have positive data and you don't have a way to put it on the market. It's going to cost you money. But when you think about how much it costs to put a device on the market, and probably Jeff could speak to that, it's not that huge an investment when you're looking at a market that's $1 billion or in that vicinity.
The way that we approached it was kind of unit operations, and where there was something off the shelf that we could use, we went that route. Or if we saw that there was something that could be automated, or as part of the strategy, then we implemented that. So it wasn't that from day one we went in for fully automated, and then for phase 2 we've automated at a small scale. So it's not gone into the commercial scale operation. So trying to stage the investment as well.
So the key to that is that the automated steps that you're doing are scalable. So when we're moving from the phase 2 equipment into the phase 3, we're actually just sizing up the movements, but the cells are experiencing the same process steps and conditions.